Okay, thank you very much for inviting me to come here and give a presentation. And the talk this afternoon will be about what we call ancient DNA. It means the retrieval and analysis of the genetic information from ancient materials and fossil remains. And it's actually a quite young research field. It's only about 20 years old, and it operates, you can say, on the interface between molecular biology, paleontology, geology, and archaeology. So it's truly multidisciplinary in nature. So I'll start this talk by giving you a very brief introduction, historical introduction to the field, and then I will continue with some of the research projects that my group has been and is involved in. Okay, so it basically all started back in 1984, where a guy called Alan Wilson from Berkeley University showed it was possible to obtain DNA from the skin of the quagga. This is a quagga. It's a zebra-like animal that lived and died out in South Africa in the end of the 1800s. And then the year after, a young Swedish student at this time, his name is Svante Pääbo, who is now one of the biggest names within human evolution. He claimed in Nature, which is this prestigious journal, that he could in fact obtain DNA from the skin of an Egyptian mummy several thousand years old, okay? And then in the end of the 80s, 1980s, beginning of the 1990s, we saw a technical revolution, which is called the polymerase chain reaction, which basically for the first time made it possible to retrieve, you can say, single surviving DNA molecules, okay? And this really revolutionized molecular biology as such, but also had a major impact, of course, on ancient DNA research. And in the beginning of the 1990s, you could read in high-profile journals like Nature and Science, well, it's possible to obtain DNA from amber entombed insect, dinosaur bones, magnolia leaves, all millions of years old. So there seemed no limitation to what you could actually obtain genetic information from, right? However, after a few years, it actually turned out that this DNA sequence from the Egyptian mummy probably came from Svante Pääbo himself. Okay? And after a few more years, well, then it turned out that all we call geological ancient DNA, it means DNA that is more than 1 million years of age, was in fact the result of either human contamination, it wasn't Svante Pääbo in all cases, I must say, and in one case probably somebody eating a chicken sandwich within the laboratory, right? So you can say quite early on it became evident that it's very easy to obtain what we call false positive results due to contamination with contemporary DNA. So why is this the case? Well, the DNA molecule, which actually creates, I should probably mention that, which actually makes us who we are, right, is actually a fairly unstable molecule. So it will decay with time if it's not repaired. And this is due to various processes, bacteria eating the DNA in the sediments, nucleases chopping up the DNA. But even if it survives those processes, it will still decay due to spontaneous chemical reactions called, for example, hydrolysis and oxidation, which will take place spontaneously in the presence of water or oxygen. And in fact, even when I'm talking right now, the DNA in my cells will constantly be damaged due to these degradation processes, and I will probably die before I reached home if it wasn't due to an extensive repair system that goes in and repair all these damages, okay? However, when an organism dies, the repair system stops functioning while these spontaneous degradation processes, they continue. So the speed by which this degradation takes place is highly dependent, you can say, on the exact environmental conditions in which the DNA is preserved. And this really, embarrassingly enough, makes it impossible for us at the moment to say precisely how long time can DNA survive under various environmental settings. Many people have tried, including myself, with all kinds of models, and every time we find out, well, we were wrong, you know. However, what we do know today is that in general, some areas are better, some environments are better of long-term preservation of DNA than others. For example, it's very good to be under frozen conditions because all the chemical reaction goes on very slowly. It's also good to be under cold conditions. quite surprisingly, it's actually not too bad to be under hot, but very dry conditions. Their degradation also seems to be slowed down. What is really bad is hot and humid conditions, like in the tropics, for example. So there we are talking about maybe a DNA survival of a few hundred to a few thousand years, while under frozen conditions, well, the record, which I got, is around 500,000 years. But I wouldn't exclude the possibility. We are working, for example, at the moment on ice samples from Antarctica, where the temperature is -50 and has stayed that low for hundreds of thousands, maybe millions of years. Maybe their DNA can survive, maybe for millions of years. Who knows? We will find out soon. Okay, so maybe we can't obtain DNA from amber and tumbit insect or magnolia leaves, but what kind of material can we in fact obtain ancient DNA from? Well, we can obtain it from bones and teeth of animals and humans. We can also obtain ancient DNA from soft tissue of animal, humans, and plants, and that includes things like hair. And it's not, you don't need to have the hair root, which is the living part of the hair. It's actually enough to have the hair shaft. It's in fact an excellent source of ancient DNA. I will get back to that later. It also contains things like ancient feces, ancient shit, which is what we call coprolites in scientific terms. And then it in fact also includes things like ancient sediments and ancient ice, where it has been proven possible to obtain DNA from animals and plants, also higher animals and plants like mammals and birds, even in the absence of any macrofossil evidence in the form of bones or hair, etc. I'll get back to that also later in the talk. So how do we do it? Well, honestly speaking, it's not rocket science. It's actually fairly simple, right? I mean, you basically, you take a piece of bone, you grind it up into bone powder, and now I didn't have any picture of bone powder, so this is cocaine on a glass plate, but it's pretty much, it looks pretty similar. Well, then you basically add some reagents, some chemicals, lysing all the cells, releasing the DNA, the genetic information, so we can get removed every other cellular parts like cell walls, proteins, etc. And then today we are using, a new technological revolution. I'll get back a little bit more into this later in the talk, called these second-generation sequencing machines, which allow us to sequence, get the genetic code out, so to speak. In massive amounts over a very, very short time. It has been, you can say, what the PCR, what the polymerase chain reaction was in the late 80s. This is what these second-generation sequencing machines are for modern molecular biology today. But we can use them in different ways, and I won't go into much details, but you can either do what is called a shotgun sequencing, what we're using when we are, for example, doing a genome. There you're sequencing randomly what you have in your DNA extract, or you can actually go in various ways and target specific regions of the DNA you then afterwards sequence. And this is, you can say, you typically end up with some kind of picture like this. This is what you see also in CSI, right? It's actually, you can say this is the order of the 4 different bases which decide that you are you and me, I am myself, right? And this is a chimp, etc. And it's actually only the order of 4 different bases together. And so what makes it problematic is that it's basically some old shit that we're working with, right? So it's highly degraded, and that creates a number of problems. There's inhibition of the polymerase chain reaction due to degradation products. There's DNA damages, and some of these DNA damages can in fact result in errors in the DNA sequence you get out, so you actually get the wrong code out, right? And then, of course, the biggest issue is contamination with modern DNA, right? So you get these false positive results. And I won't get into major details, but let me just put it this way. We're completely paranoid with contamination. So, for example, we are using these full body suits, face masks, special labs with positive air pressure and nightly UV radiation to erase all the DNA, right? And these labs are separated physically from any other modern biological lab, in order to reduce any contamination, provided by the researcher himself or reagents, etc. And obviously a huge issue in terms of contamination is what we call sample-based contamination. It's these archaeologists that have never heard about DNA research, right? So they don't use gloves or anything. They just handle the bone, you know, and putting the DNA all over it, right? And that's the reason why modern excavations that we are controlling, they're looking like this. I mean, this is, you know, in full body suits, face mask, gloves, etc. This is in fact an excavation I was part of in the northernmost part of Greenland a couple of years ago, where we tried to find out who is the earliest people who settled in the Arctic. So we know that people have been present in the Arctic for around 5,000 years, but I mean, it has remained a mystery who was these first people. Are they, you know, ancestors of present-day Inuits or Eskimos, if you want, the guys that are actually inhabiting this area today? Or is it Native Americans, Indians, or is it something completely different? And one of the big problems was that although you found, or you find, you can say, you know, the house remains, the tools all over the Arctic, you just don't find any human material. I mean, in terms of bones and graves, etc. Nobody knows why, but maybe they buried, you know, the dead on the sea ice or something, right? So the idea here was originally to go up here to find some of these sites, where archaeologists have never been digging before, so there was no contamination. And then my idea was, well, people have sitting handling, you know, the tools, etc., putting the DNA on it, right? So maybe if we excavated those tools under very controlled conditions, we could actually go back home in the lab, extract DNA from the tools and found out who they were, right? And I came back with a massive amount of tools, and there was just no DNA in it. I mean, well, the bone tools showed the animal DNA, but no human DNA. And I actually thought, well, this is a dead end. We'll never find out who they were, but we did in the end, but in a complete different way. I will get back to that in the end of the talk. Okay, so what kind of questions can you then address with this ancient DNA? Why are we going through all these problems, you know, in order to get this DNA code out? Well, one of the things that my lab has been really interested in is what we call the late quaternary megafauna extinction. It's basically the extinction of big mammals in the end of the last ice age. So if you go back just 10, 12,000 years and look at what was the environment like in Europe, Asia, and in Americas, you would see a world very, very different from today. And one of the things that made it very different is that at that time you had a massive diversity of big mammals. So you had elephants in the form of woolly mammoth. You had rhinoceros in the form of woolly rhino. You had lions. You had giant deers, bison, giant ground sloth, etc. So what you should imagine is something similar to an African savanna today, but under Arctic conditions. So there was literally hyenas and lions running around in the snow. Okay. For some unknown reasons, the majority of all these big-bodied mammals died out, presumably around the end of the last ice age. And this is a massive extinction. I mean, it's probably what we can call the last, the last major extinction on Earth. We're talking about around 2/3 of all these big-bodied mammals went extinct of the species. And scientists obviously have been very puzzled with what's the reason for this massive extinction. And there's been proposed a number of different theories. Some of the most popular ones are that, well, it was humans. We know that our ancestors, modern humans, came out of Africa around 80,000 years ago, spread to the rest of the world. According to this theory, they met these big-bodied mammals that had never seen humans before, so they were just standing, what is this? While the humans basically slaughtered them one by one. And according to this theory, it happened very, very quickly. It has to have happened very quickly in order for the theory to make up. So you're talking about it happened in a blitzkrieg-like manner. It's an expression from, I think, World War II, something about coming in and hitting the enemy hard and good very quickly. So people say, well, it happened within 1,000 years after first contact. This is a theory that is super popular in the States, for example. Surprisingly enough. Okay. Then some people say, well, it's impossible that a fairly few amount of humans with a primitive technology could kill off all these mammals. I mean, this is just, we can't imagine that. obviously it must be climate change, because we know at the end of the last ice age, the climate became warmer and more humid, and according to this theory, well, it changed the vegetation cover and the whole ecosystem collapsed, right? Then, well, the critics of this theory says, well, okay, what was so special about this last, this climatic event? I mean, these guys have survived climate changes before. What was so special? And then the climate people say, well, we don't know really. Okay. Then, more recently, there came a theory where some geologists argue, well, there was a meteor that was basically hitting North America 12,900 years ago, and that created a number of wildfires and even initiated a climate change, and that caused the extinction. But people are still discussing, even today, was there in fact, an impact or not, right? So it's not kind of completely certain. And what I should mention is this, I think, the reason why I think this discussion is really interesting is not only because, well, it caused the extinction of a lot of big-bodied mammals, but also because it's actually, which of these theories, if any, that is correct, will have a major impact of how we look at, you can say, at our environment, right? For example, if the climate theory is correct, well, then we must say, well, climatic changes as we're experiencing at the moment can have massive impact on the biology, right? I mean, that would be the conclusion. Then we actually should be very worried about what is happening at the moment. If Blitzkrieg theory is right, well, the next time you see a Native American from South America sitting in the television saying, oh, please live, with nature like my ancestors already done, you can just say, right? I mean, your ancestor fucking killed off the whole megafauna. I mean, so it actually has quite a big influence of how we look at things. Okay, so some years ago I was involved in a study where we tried to test this idea using ancient DNA. So what we did was we used the bison, the steppe bison as it's called, as an example. So we went to China, Siberia, North America, collected hundreds of steppe bison bones. We extracted DNA, mitochondrial DNA as it's called. It's from the mother line only. We extracted it from them. We also dated each of these bones, which you can do with what is called C14 dating. And this massive data set, now you're not used to look at these trees, but that tells you a lot of different things. And one thing I just want to very briefly mention, just as a side note here, is that if you, for example, everybody knows the plain bison, right? The one that the Native Americans were shooting from the horses and Buffalo Bill killed like several hundred a day, etc. Okay, so today, the genetic diversity in the plain bison is tiny, right? I mean, basically each bison you take is nearly identical because everybody thought, well, this is because the Europeans came to North America where there was millions of bison and then they were shooting off the whole thing, right? So there was only a few hundred left in the end of the 1800s. And this is basically bison skulls from that time, right? So they were just shooting them from the train and from the horses. They shoot everything they could look at. And therefore they said, well, I mean, they went through this massive bottleneck and therefore they have this very little genetic diversity, which a very low genetic diversity normally is the same as being very vulnerable to any kind of change, okay? Well, when we look at the data, we can see that the plain bison have had this very low genetic diversity for the past 10,000 years. I mean, it has nothing to do with the off-kill of the bison, okay? So basically what it shows is that the bison went through another bottleneck for whatever reason around 10,000 years ago, and that actually created that nearly every individual is identical. Okay, what else can you do with this data set? Well, what you can do is you can actually estimate what we called the effective population size. And popular speaking, it's just the number of animals through time, okay? And you can do this with all kind of mathematical modeling, but it demands a lot of sequences and a lot of dates, but we could do this for North America. And what we can see is that the number of bison individuals kind of increase around 100,000 years ago. They increase. Around 25, 30,000 years ago, suddenly, bang, the population just goes down, I mean, right? And if this is correct, this is really important because that shows us that most likely humans had no involvement in this crash, because the oldest evidence of humans present in the Americas is around 10,000 years later. Okay, then when this crash actually happened.
What is the red line and the blue line?
Okay, this is confidential intervals. It's basically the uncertainties, right? So you should look at it like a thick thing. I won't show this, but we are finishing a massive study where we looked at woolly rhinos. We also looked at big-bodied animals that survived, like the reindeer. We have looked at muskox that just barely survived, etc. And we have not finished analyzing this data, but what it do suggest is a really complex pattern. I mean, it's not like you can say it's only climate, it was only humans. It actually seems like for some animals it's probably humans, but for others, I mean, humans have nothing to do with it. So we'll get that out soon, hopefully. Okay, another thing that we are interested in is reconstructing past ecosystems. So you can say, I mean, two major limitations in ancient DNA work until recently was one, that most fossil remains do not contain any DNA because it's all degraded away, and the other one obviously being that many fossils are really rare, right? I mean, you might go out and find a lot of mammoth bones when you're walking around in the tundra, but looking for a sable-toothed cat, for example, I've been there, I mean, numerous times, I've never found one, right? So therefore it wasn't really possible, you can say, to reconstruct entire ecosystems, because in a spot like this, for example, say, how was it 20,000 years ago here? I mean, well, if you don't find any bones, well, you couldn't say anything about it was like here, right? And that was what initiated the idea that maybe we could go and look into sediments, ancient soil, directly, and then recover the DNA from these animals and plants that had been living there thousands of years ago. And the rationale here being, well, if I do like this on the table, I put my DNA on the table, right? We can't see it, but it's there. The question is, how long time will it survive, right, through time? So in order to test this idea, we went to northeastern Siberia, to the permafrost regions, because here you have an ideal environment for long-term preservation of DNA. It's permanently frozen soil, right? So if there should be anything to this idea, well, it should definitely work in Siberia. I must say, at this time I was a PhD student, and I remember my supervisor said to me, this is the most stupid idea I ever heard. But anyways, we did it, we went there, we did it, and I tell you, we got very, very surprised, because it turned out, not only from Siberia, we also took sediments from caves, non-frozen sediments from caves in New Zealand, and it turned out that in 2 grams of sediment, which is basically the same as a cubic of sugar, we could find a huge diversity of those animals and plants that had been living in these regions through time. So in Siberia, we found DNA from woolly mammoth, reindeer, muskox, horses, etc. Also small animals like hares and lemmings, as well as numerous different plants, right? And in New Zealand, we found DNA from different species of extinct moa birds. It was just like, I mean, this is amazing. And we also got it published in Science, and obviously this was a major breakthrough at the time, because it really, in principle, show you can go out to whatever region, even though there's no bones preserved or anything, and then you can reconstruct what plants was there, but also what animals was running around eating these plants, right? And what is then the reason why you have all this DNA, right? And that was something, of course, we asked ourselves already then and been puzzled with ever since. But I would say what it seems to be the case is that for the animals, it's actually DNA coming from the urine and feces of the animal. Yeah.
Why can't you then find in the Arctic just take sediments from the ice or whatever, and then find the proof of human beings?
It's a very good question, because it's all a matter of concentrations. Okay, so I mean, we have actually tried going to some of the human sites, but I mean, there's a limit. You can say you need a certain amount of DNA in order to, both for it to survive through time, but also in order to detect it. I mean, there's no doubt it's there, but it's probably, maybe it's very scarce or whatever. You have to take a lot of samples to detect it, right? Or there's simply too little to detect it. I only tried it once though, so I mean, we'll see.
You have to go back.
Yeah. But anyways, it seems like it's feces, right? Because when you basically take a crab I mean, there's DNA from there's cells from the intestines coming out containing DNA, and the same with the urine. And for the plants, it seems to mainly be these what we call fine rootlets that are less than 2 millimeter in diameter that are constantly being produced and shed by plants into the sediments. And there is evidence showing, well, when the cells enter the sediments, the cells will very quickly be opened, the DNA will be released. Most of this DNA will be eaten by bacteria or degraded. But because DNA is charged and sediment particles are charged too, the DNA will actually bind to these particles and in that way survive over time. At the moment, this is speculative, but we're actually testing whether modern bacteria can then also incorporate. I mean, bacteria can take DNA from the surroundings, incorporate it into the genome, and we're testing whether they can do that with ancient DNA as well, whether that's a way to get long-term DNA survival. Okay, what kind of questions can you then use this technique to address? Well, one of the things we're interested in is when did these big-bodied animals, these Ice Age mammals, when did they actually die out? Huh? Sorry? Oh, okay.
No, I just missed the word, sorry.
Okay. Okay, cool. When you can read in the textbook, for example, well, woolly mammoth died out in mainland Siberia 11,000 years ago or whatever, what is that actually based on? Well, that's based on people like me running around in the tundra, picking up mammoth bones, right, bringing them back to the lab, do a C14 date, which is really expensive, you know, and then sometimes if you date a lot, then sometimes you get a young date, and then you say, okay, this is probably a time where mammoth got extinct in this area. But then you can ask yourself, what is actually the chance of finding some of the last survivors of mammoth using this approach, where you have a preservation of bones over hundreds of thousands of years, right? And you can't, just by looking at it, see, well, is this mammoth bone 11,000 or is it in fact 30,000 or 50,000 years old, right? So I got some statisticians to look at this for mainland Alaska, which is one of the areas where you have done most mammoth dates in the world. And then they basically came to the conclusion, well, if you actually had a driddle population towards the time of extinction, let's say it went down by 30%, well, the chance of these guys, even though you are collected bones, not finding, some of the late survivors even surviving, several thousand years after the youngest bone date, is more than 50%. In other words, I mean, this is a fucking useless approach, right? So what I then thought was, well, when a mammoth die or another of these big-bodied animals, obviously, it only leaves one carcass, one body behind, right? And that's what you're trying to find. It's very difficult, right? It's searching like a needle in a haystack. But what about how much does a mammoth actually pee and shit during its lifetime? Well, nobody knows, but a modern Indian elephant pees 50 liters of urine a day. It means it's DNA all over the surroundings, right? And crapping all over the place. It means it basically put marks all over the landscape, and obviously it must be much more easy to find those marks than actually go and find the body. And that's exactly what we did from central Alaska. We went into this called Stevens Village. There's a very well-described permafrost record. It's very neat, become younger and younger through time. There's no problems, no inversions or anything. And then we just started looking, okay, what kind of animals do we find in the different layers of different ages? And mammoth and horse we found in layers that are at least, that are 10,500 years old. And when we look at what is the youngest evidence of mammoth, for example, in mainland Alaska, well, that's 13,100 years. Then you can ask yourself, well, does it really matter that we find something that is around 3,500 years younger, than the youngest bone record? Yes, it does. Because if you look at the oldest evidence of humans' presence in Alaska, well, that's around 14,000 years, right? If you compare 14,000 years to 13,100, it's 900 years difference. It's perfect Blitzkrieg scenario. And that's obviously one of the reasons why the Americans just love that idea. However, when we actually suddenly push the date of mammoth extinction back even 3,500 years, that means that mammoth and mammoth have coexisted for several thousands of years before they went extinct, and obviously it cannot have happened in a blitzkrieg-like manner. The same for the impact hypothesis, with this impact that hit North America. They should all have died out 12,900 years. No, they didn't. I mean, they survived. In other words, there might have been an impact, but it didn't kill off the mammoth, okay? And this is the kind of approach we then are doing at different places in the world. You can even take this dirt DNA, as it's called, a step further by applying it to these deep ice cores that have been drilled all over the world. And Denmark is actually one of the really great ice core drilling nations. So, people are drilling ice cores because they want to use them for studying climatic changes through time. Because ice is, in fact, it's basically old snow that has been falling down, and then under pressure, it turns into ice. So you have kind of a climate record through that. And most of this ice look like this. This is what we call clean ice. I mean, if you melt it, you can drink it. There's no problem. I mean, even though it's 50,000 years old, it's like mineral water. But when you hit around the bottom of the ice cap and the ice cores, you get what is called silty ice. It means it's ice that contains a lot of dirt in it. And suddenly it hit me, well, maybe there's DNA preserved in this dirt, and we can find out what kind of animal and plant communities were present in places like Greenland and Antarctica the last time these areas were free of ice. And this is something you have no idea about. And maybe you'll get surprised to hear this, but it's in fact 10% of Earth's terrestrial surface, even today, that is covered by thick glaciers and ice caps, and we have absolutely no idea what was present there before glaciation, okay? So this was, of course, interesting to check out whether it would work or not, and we tested 3 different cores, 2 from Greenland, 1 from northern Canada, from this silty ice. And, you can say that this one from the Greenland summit, that didn't work. It's not maybe too surprising. It's quite warm in the bottom there, and it's believed to be very, very old. The John Evans Glacier was kind of a positive control, because that one from Canada, because that glacier is only 4,000 years old. So we should expect to find some of the same plants and animals that is around that area today. It hasn't changed too much, and that's in fact what we did, so that was perfect. The big surprise was from southern Greenland, southern central Greenland, because what we found evidence of here was a diverse conifer forest. Nothing like you see in Greenland today, with spruce, pine, taxus, birch, various types of insects. And we were just like, fuck, I mean, this is amazing. I mean, there's a forest there. And you can say the youngest evidence of forest in Greenland at that point was 2.5 million years, okay? We was just like, what is the age of this thing? And there we went into really troublesome water, because people doing modeling, right, of the Greenland ice sheet in order to predict what will happen in the future, many of these modelers said, well, southern central Greenland lost all of its ice during the last interglacial, the last warming period, where the temperatures in Greenland were 5 degrees higher than today. And this massive flow of ice into the ocean created a global sea level rise of 4 to 6 meters. And obviously that's what everybody is really afraid of should happen again, right? If the temperature increases and all this shit is coming out in the ocean, they will just be paddling around in the house in Holland. Okay. But then other modelers, just a minority, they said, well, we don't believe that is the case. Obviously it shrank, the ice cap, but it wasn't that extensively. There was still ice. So we were sitting here, okay, we have no idea what is the age of this forest. When was this area last free of ice? And in order to attack that problem, we did the first direct dating of this silty ice. The problem is, when you look at the clean ice that they use for climate change, it's like tree rings. You can count, I'm here in time. But the silty ice can either be just a little bit older than the clean ice, but it can also, in some cases, be much older, right? So this was the question. We did 4 different dating approaches, and I won't go, the molecular clock, some amino acid ratification exposed something saying something about the age of the biological material in there. Then we used 2 others, buridium chlor dating, which says something about when was the inorganic particles, all the soil when was that last exposed to the atmosphere, and OSL dating says something about when was this inorganic material last exposed to sunlight. And the thing is, you can see there's big arrow markings, but they all show it's much older than the last interglacial. In other words, southern Greenland, southern central Greenland wasn't free of ice during the last interglacial when the temperature was 5 degrees higher than today. In fact, where they overlap is around 450,000 years ago, which is the previous interglacial. I tell you, when we published this stuff in Science, I mean, first we had a reviewer, right? I mean, at that point, the climate debate was already really, really hot, and Bjørn Lomborg, he was kind of out, and the climate believers were in. And we came here and said, well, maybe the ice isn't as unstable as people thought. One of the reviewers just said, I don't believe it. I don't believe it. And first we had 2 dates, then we took 2 more dates. He still said, I don't believe it. So in the paper, in order to get it into Science, which we obviously really would like, we basically had to write, well, this is what we find, but we don't really believe it. But obviously we do believe it. I mean, this is 4 different independent methods basically showing the same thing. And then the year after our paper, there came another paper in Science where they looked, on a marine core outside southern Greenland, got exactly the same result, okay? However, then it hit the media. And I tell you, I mean, this was my first paper, and well, it wasn't the last, but unfortunate, but it was the first where I actually went into this climate debate, right? And I mean, you don't believe how terrible it was. I mean, we made this press release, right? Then journalists had just picked words, around in this press release, sticking it together. So if you took a right-wing paper, it was like, yeah, you don't have to be afraid of climate temperature increase anymore. Scientists have just shown that. If you took a left-wing paper, it was like, oh, fuck, we're drowning tomorrow, right? and I got emails. I remember that even the Danish media, they ran these stories, and I had to go into TV2 News and basically say, well, I have to explain myself here. And then I explained the journalist, I mean, maybe even though that the ice, didn't disappear from Greenland, I said, well, we are not questioning that there was a sea level rise. It's just that there's something wrong with the models, that the water must have come somewhere else. And I'm sure, that Mrs. and Mr. Denmark don't care when they're sitting in their rubber boats, from where the ice comes from. But it's really important from a scientific point of view. And then the journalist said, oh yeah, I got it, And then they started, the news. And he was like, yeah, okay, so that's positive news. Nothing will happen. Is that correct? I was just like, fuck, I mean. And I started getting emails, from people saying, well, you have the DNA of Donald Rumsfeld. And then another one saying, well, oh, I'm so glad to hear about your paper. I always said to my grandson, there's nothing to be worried about. I was just, fuck, I mean, it was crazy. I mean, it was just crazy. Anyways, that might, what southern Greenland might have looked like 450,000 years ago. Okay. Another thing that you can do with ancient DNA is what we can call ancient functional genetics, which is basically using the genetic information to say something about either how the animal or human looked or about the behavior directly from the genetics, okay? And I was involved in the first ancient functional genetic study, and that was in fact on the woolly mammoth, because sometimes you find a woolly mammoth mummy with, fur on it, muscle tissue, and even hair. But they're always red-haired. I mean, even if you go to the Zoological Museum, you will see one of these reconstructions. You see this red-haired elephant, right? And then you could ask yourself, were all woolly mammoths actually red-haired? I mean, if you look at many other mammalian species, just look at ourselves, there's all kinds of hair variations in color, right? So we said, okay, we want to test this. Maybe it's just a degradation phenomena on the hair. So we sequenced what is called the MCR1 gene of a mammoth bone, and we know that the MCR1 gene is really important for what kind of skin and hair color a mammal will get. And we could see in this one mammoth bone, there was in fact different versions of this MCR1 gene, right? And this was, where the versions had substitutions which were very significant. And what you then can do is you can actually put it into an expression vector and then express the vector in a mouse. And then the mouse will in principle get the same hair color as this version is accounting for, right? So there we could see, well, in this one mammoth bone, you actually, one of the versions, well, that was completely blonde another version was completely black. And obviously, in a population of woolly mammoth, there will be individuals only with one of the versions, others with only the other version, and then obviously some with a mixture of the versions. So most likely the woolly mammoth have ranged in color from completely blonde to completely black and everything in between, okay? And then, one of my colleagues, Clovis Fox, actually took it a step further by saying, okay, what about the Neanderthals? Because Neanderthals, don't find any soft tissue. The only thing we know from Neanderthals is our closest relative. I mean, the chimp is our closest living relative, but if you also include the dead ones, well, then it is the Neanderthal. And they died out. It's this archaic human that died out around 30,000 years ago. But you only find the skeletons, right? So people have speculated, what did a Neanderthal look like, right? And so Clovis Fox, he took the MCR1 gene from Neanderthal bones, expressed it in mice, and found, well, they were all reddish hair with blonde reddish hair. So they were kind of prince-like, and maybe not these like these kind you see in the what is called Kampmamillen in Danish. This. Anyways, then Svante Parbo, who at that time had recovered over his mummy failure, he actually he said, okay, what about, could the Neanderthal actually speak? Because this has been a major discussion. Some people believe that the reason why the Neanderthals died out while our species survived was that the Neanderthals couldn't speak, so they couldn't communicate properly with each other. Otherwise, the Neanderthals, they had bigger brains than we had, and they had definitely bigger muscles, right? So they're much stronger. There must be some reason why they couldn't survive, right? And then he sequenced the FOXP2 gene, which is a gene we know is super important for the ability to speak. There's only two base differences between us and the chimp, but the chimp can't speak. And in fact, there's some children that are born with one of the chimp versions, and they have really big difficulties speaking. And he found, well, the FOXP2 is identical to modern humans. In other words, Neanderthals could most likely speak. And it was actually quite interesting. I met with him up on a conference about a year ago, and then he told me, well, we also took this human version of the FOXP2 and spliced it into a mouse, and then we, as he said, well, then I start trying to talk to the mouse, and it said peep peep, so okay, that not really work. And then, but then they studied the behavior by putting it into a room, and trying to study, is there any differences? And apparently a modern mouse will kind of just running around in a room exploring, right, very unfairly the room. This human version, right, was just climbing along the walls, So it apparently also have kind of an effect on the behavior. Okay. Another thing that my group is very, very interested in, has been and still are very interested in, is the early peopling of the Americas. So we know that the Americas was the last continent to be where people came in. Okay. However, when it comes to when did these people come in, from where did they come, what routes did they come of, there's no consensus whatsoever. You have absolutely no idea how many theories there's out there. So there's, of course, the classical one you can read in most school books. Well, they came from Siberia over a land bridge, and then in Alaska, they met this massive ice cap that was lying all over North America, and they had to sit waiting in Alaska until the ice-free corridor formed, and then they were going through that ice-free corridor several hundred kilometers, and when they passed it, they killed off all the big-bodied animals, and they developed what is called the Clovis technology, which is these massive spear points, etc., which is very unique to America. But there's very high estimated researchers in the Americas, for example, that say, well, the first people that came in, Clovis, in this site Clovis, was in fact Europeans. They came across the Atlantic, and then they were living in the Americas. Then at some point, the Native American ancestors came in from Siberia and wiped them out, okay? There's even people saying, well, no, they came from Australia, or they came like to higher, they came from Africa, sailing on some kind of boat. So there's all these theories out there. But the thing is that if you look at the facts, well, at that point, the oldest directly dated human remain from the Americas was 11,500, what we call radiocarbon years. It's when you date it with C14. In calendar years, in normal years, it's around 12,300. It's just because the calculation, there's some differences, you know, depending on the model. So if you want to be very precise, you do it in radiocarbon years. The oldest generally accepted occupation site, they found no human remains, but they found some tools and stuff, is what is called Monteverde in Chile, in South America, around 12,500 radiocarbon years old, okay? This is basically the facts. Okay, I got into this stuff because I was contacted by this dude here. His name is Dennis Jenkins. He's an archaeologist. He's doing excavations in southern central Oregon in some caves here called Paisley Cave. It's kind of a desert-like area, these caves. And he's called me and said, well, you know, this is an amazing place. I found, you know, the bones of these extinct animals like camels, etc., horses in the Americas, and some of them even have skin on them. So it's that well preserved. But additional to that, I literally find hundreds of ancient coprolites of these ancient shit, right? And he said, well, I'm not a specialist, but when I look at some of this shit, you know, it looks pretty human to me. And could you please find out whether it's a human poo or not? And first, honestly speaking, I wasn't very interested because I thought, well, some kind of Native American and taking a crap in that cave wouldn't really bring us much further, right? But on the other hand, I said, okay, maybe this could be a way into this kind of research and then do some more interesting stuff afterwards. So I went to Oregon, took like 14 samples of these coprolites and brought them back, and they were in fact lying in the freezer in my lab, you know, for 1.5 years. I didn't do anything with them. And then one day a young Spanish visiting student came by. She should be there 3 months. She didn't have a project. She said, oh, Professor Willerslev, what should I do? And I said, look at the shit in the freezer. And that's exactly what she did. And then, you know, a month after, she came back and said, oh, Professor Willerslev, I'm finding Native American DNA in some of these feces samples. And you can say, well, that's not too surprising. But when we looked at the position in the stratigraphy, they could be very old. And obviously, then I became really interested. And in fact, she was shipped home. So obviously, there was 2 critical questions we had to address. One was the shit in fact as old as suggested from the stratigraphy? That was quite easy to deal with because this is organic material. We sent it to 2 different C14 dating labs. They come back and said, well, I think it was 3 or 4 of them are in fact 1,000 years older than Clovis. Okay, so this is good. Then the next question was, obviously Dennis Jenkins had no conception of DNA work. So his people had just like, oh, this is the shit sample, right, without gloves or anything, down in the back. And who knows, I mean, some of these guys could be Native Americans, even without knowing it, right, on the mother line. And so this was, I mean, they could have put the DNA there. So we really had to deal with that problem. So I said to him, well, we need you to contact every single person that has been participating in these excavations. I mean, they're going on for 2 years. It was 58 people. And then we asked him to, each of these people took a piece of hair, put it in an envelope, sent it to Copenhagen. and we typed them. And in fact, one of them was Native American, but a different genetic type than what we found here. So this seemed to be true, you know. There's different, what we call haplogroups, different genetic types. And this had really big implications, you know, for early peopling of the Americas. First of all, Clovis' first hypothesis, forget about it. There was people before Clovis, around 1,000 years before Clovis at least. The migration as traditional, you know, in the school books, where it's coming through this ice-free corridor. As I said, they came here from Siberia, and then there was this massive ice cap, and at some point they formed this corridor, and they went down here. It cannot have happened for the first ones, because the ice-free corridor wasn't open at that time. It's much more likely that they came along what was called the coastal route, the western coast of Europe. It's the oldest directly dated human remain, similar in age from the Americas, similar in age to Monteverde, around here down in South America, which is nice. You know, it's where you have dated some tools, but where people are still discussing whether it's valid or not. Because if people should come this way, right, it's fucking odd that you find the oldest spot on the southern tip of South America. So this is really good. What we also could say is this is haplogroup B, I mean the oldest of them, which is a Native American haplogroup. In other words, it seems like that these guys, at least on the mother line, are in fact ancestors of present-day Native Americans, okay? In some kind of version. It's definitely not Europeans or anything like that, or Australians or whatever has been suggested. And why is this important? This is hugely important. Because what is the reason why all Native American tribes today, for example, claim all these ancient skeletons that are dug out of the ground, right? They basically say, okay, it's our ancestor, come with it, we don't want science to look at them, and then they rebury them, burn them off or something. This is because, I mean, they are super afraid that someone should find evidence that there was, let's say, Europeans in America before Native Americans. Because they have a lot of rights, for example, to casinos in states where you know, gambling is not allowed, because they are what they call First Nations, right? If suddenly you came and said, well, First Nations, my ass, I mean, there was Europeans here, they just came back and took what was rightly theirs. Obviously, I mean, that would create hugely problems. So, you know, different to the ice core story, which was, you can say, political incorrect, this one actually landed very political correct. And of course, this was really nice because, I mean, in Denmark, you have no idea of what is on stake here. I mean, people, are suing each other like mad, you know, almost killing each other in order, you know, to basically, the fight between scientists, between each other, but also between Native Americans and scientists. And I want to mention one more thing. We have in fact, oh no, this is on video. I won't mention that anyways. Sorry, I don't want that to. Well, let me put it this way. There exists one Clovis skeleton. I mean, the reason why people have kind of said, okay, Clovis is Europeans or Clovis is you know, Australians or whatever, is that if you look at the skeletons, the very early skeletons in the Americas, I mean, even I have no idea about, you know, skeleton morphology, can see, well, this is looking completely different from a Native American. I mean, these guys are in fact looking like Neanderthals, not almost. I mean, they have eyebrows like this, right, and a forehead like this, right? So I mean, they are really, really archaic. There exists one skeleton which is found together with Clovis remains. And while most of these other old skeletons in the Americas, you know, the native tribes have actually reclaimed them and reburied them, etc., burned them off or whatever, then this skeleton, you know, is private owned because, you know, in the States, it's not like in Denmark where you have this Danekrig, right? I mean, if you find something on your land, it's yours. and they've digged out this Clovis skeleton, and, you know, well, that has been kept with this family, Antic family, and I heard about it. I contacted the family and said, do you guys want to go to Denmark? You can bring your nanny, you can bring your boyfriend, if you just bring me a little piece of that old skeleton. And they did, and we are in fact testing it now, you know, and we have some results, but I can't show that on the video because it's exciting. It's really exciting. Okay, you can actually... No, it's okay. It's well, no. I mean, I will be killed if I say something about this, anyways. I mean, can't spread the rumor right now. I mean, it will soon come out, I think. You know, okay. So I mean, obviously, when you're doing this shit on other people, you should also look into yourself and your own history. I mean, otherwise you'll just be said, well, you're sitting there, you know, changing our history, right? What about yourself? That's exactly what we did last year. We said, I mean, in Europe and Scandinavia as such, you could say some of the, I would say one of the probably the biggest questions in archaeology is what we call the neolithization. It means the transition from hunter and gatherers to farmers, okay? And there people are really also fighting, I mean, almost killing each other about who's right. Because the thing is, if you look at Europe and Scandinavia, most places in Europe, well, we know that the first people that came in after the ice disappeared after the last ice age were living as farmers, hunter and gatherers. And one day, it seems to be almost one day, you know, they put down the bow and arrow and started, you know, milking cows and, you know, cutting hay, etc. And people, of course, have been puzzled with, is this, I mean, just a cultural transition? Suddenly they just found out or learned from somebody else, well, it's smarter to milk a cow than go and kill a deer. Or was it, in fact, another people coming in? And obviously, this has a major impact, for example, how long time back you can actually trace us into where we are today, right? In order to address this question, I would say that Denmark, in fact, has the most unique skeleton collection in the world, because there's a lot of skeletons both before, this transition and just after and in between. The problem is with Danish archaeologists, I'm not sure whether there's anybody present here, but I mean, they're just fucking fighting each other. I mean, it's crazy. So, I mean, we can't use a single skeleton, right? I mean, we need a number. I mean, we need 20 or something in order to say something useful. Okay, 5 minutes. Okay, I will hurry up. Okay. So I mean, and in Denmark, it's like, okay, if you are working with John, I mean, I won't give you my skeleton. If you're working with Brian, you're not getting mine, So in the end, it was just like, forget about it, and we went to Sweden. And there they also have they actually have some of the what we call last hunter-gatherers, and we DNA typed them. And already when we looked at the DNA, we could see, well, this is really not very Scandinavian, But obviously, there's gone, several thousand years, and, the frequency of different DNA types can change through time. So we simulated. You have to simulate and said, okay, we took the modern Danish, Norwegian, and Swedish population and basically asked a simple question: could these guys be ancestors to us today? Right? And the answer was just like, no way. No way. Then we did it for the Sami people, which people also have suggested that the first people coming in was Sami. And then, later our ancestors came in, pushed the Samis up north, right? We came in with our plows, etc. And the answer was even more, no, there's no way they can be ancestors to Sami. Then we asked the same question for Eastern Europeans, and the answer was just bingo. Okay, so basically, and then a German team did around the same thing, in mainland Europe, got the same answer. So basically what it do suggest is that the first people coming in Europe is in fact what we today call Eastern Europeans. At some point, our ancestors came in with, with our cows, etc., kicked the butt off of these Eastern Europeans, and in fact only in what we call Eastern Europe today is a really, cultural transition, where the guys put down the bow and arrow and start milking cows, right? In the other case, it was actually a population replacement. And I think we should remember that when the Polish workers are coming back to Denmark and are being mistreated, because in fact they're only coming back home, So, okay, I have to really hurry up here. Okay.
Very quickly,
yeah.
You obviously, I'm sure you must have analyzed your own DNA. So
Oh yeah. We are doing that. It's a standard.
Where are you from?
Well, I'm at just a very boring Northern European, unfortunately. I think I'm haplogroup H, which is something very common here in Denmark today. On the mitochondrial side, I mean, obviously, I really hope to be Native American or something, but I mean, it's just it was pretty, unexciting. Okay, so everything what we have talked about now, basically all the results except this mammoth study with the hair color, is what we call mitochondrial DNA. It's basically DNA. I mean, it's like if you have a cell, you have the nuclear DNA where 50% is coming from your father, 50% from your mother. Then you also have what is called the mitochondrial DNA, where everything comes from the mother. It's in fact when the sperm penetrates the egg, then the sperm flagella contains all the male's mitochondria, but it's losing that, okay? What is the reason why we have been working with mitochondrial DNA? Well, this is because we weren't able to get nuclear DNA out. But is that a problem? Yes, it is a problem, because the mitochondrial DNA from the mother is only 16,000 base pairs long. It's a very small molecule. It really don't tell you very much about anything, really. While the rest of the genome, which makes me, you, that's up in the nuclear coming from your father and mother, and that's 3 billion bases, okay? And then, with these new, and now I don't have time to go into detail, but when these new sequencing machines came, that suddenly allowed us to massively sequence, get massive amount of sequences out in very short time. Obviously, several people, including myself, got the thought, maybe we can create a complete genome from an extinct organism, right? Like people have done, the human genome 10 years ago. And, okay, so my problem, well, I had several problems actually. I had a lot of problems because, I mean, I was not the only one getting the thought. I mean, there was actually a group in Germany, there was a group in America that got the same idea. Well, the only difference between us was that they had shitloads of money and I had nothing, okay? So I went to different grant bodies and said, oh, please, I have this excellent idea, and it was just like, I was stupid. I mean, you can't, first of all, it can't work. Secondly, what will you use it for? Typical Danish answer, right? So I was running around there. And at the same time, obviously, I needed a sample that was really good to do this stuff on, right? And there was this funny story because, see, as I told you in the beginning of the talk, I went to northern Greenland trying to find out who is the first people who settled in the Arctic, right? And I came back with nothing, or got no results. And I basically just dropped the idea. Then I'm sitting talking with Morten Melgaard from the Natural History Museum on a completely different projects on reindeer. And we start talking about this. And his father was one of the archaeologists who basically uncovered that people had been present in the Arctic much, much longer than previously believed, And he said to me, I'm pretty sure, at the Nationalmuseet, at the National Museum, that there is a massive pile of hair from one of these first individuals. I said, no way, there's no, because I went to the National Museum and I asked the archaeologists and they said, oh, we have nothing. We have absolutely nothing. And then I went to northern Greenland, spending $500,000, freezing my butt off in 1.5 months in order to find something. And I didn't find it. But he said, no, I'm sure. And then we called, and it was like, oh yeah. Oh yeah, in the basement, well, there might be something. And it was not just a pile of hair, it was a massive pile of hair, right? And we dated it. We dated it, perfect 4,000 years old, was found in the culture context of the first people into the Arctic, also dating to that period, perfect sample. So now I had my sample, It was the sample, obviously, we should use to do the first ancient genome. Then my problem was money. That was the first problem. But in the end, it was an amazing story because, I was going to all these grant bodies, getting no. Then I'm writing to, to the director of this fairing concern, this medical concern, right? I heard they had a foundation and stuff, and I asked, oh, have you a foundation I can apply for? And then he wrote back, no, we've closed the foundation, but I think your project is really interesting. So from my personal account, if you give me your account number, I can give you 250,000 to start with. I was just like, okay, okay, that's great. Okay, thanks. And then, next day they were transferred and he was just like, okay, go ahead. You can start on this, make some applications or do some experiment. Yeah. And that actually gave us the money to do the first test that I could use to go to Lundbeck, the Lundbeck Foundation, and I could show, well, this is in fact feasible, right? We can do this. And they said, obviously, this is super cool. My only problem was obviously that the Germans and the Americans, they had the last 2 years doing this, right? And they had 30 million kroners each. But then, we just have to be faster and more clever. And one of the things was that obviously, we had some of these machines, like most labs have, we had 4 of these machines, most labs maybe also had 4 or 6 of these machines. But I needed to go really fast. With these machines, I would never beat the other guys because they were so far ahead. And then, I contacted this guy, Wang Yong from China, and he heads the largest number of these sequencing machines in the world. At that time, they had 120. So I said to him, what about joining forces here and, we can do this genome together. And first he was like, it's not very interesting, etc. But then we, ate some boiled dog and drank some rice wine, all night. And he became more and more interested in this project. And in the end, we said, okay, we're doing this, okay? And this is the great thing about the Chinese, because I went to some other sequencing centers, let's say in England, in order to get machine power. And it was like, yeah, put in an application, you can put in an application and then we'll go through board 1, board 2, board 3. I could just see, fuck, it's like 6 months ahead before you get an answer. Here, you just say, okay, deal, and the next day, you're just running, okay? And that's exactly what we did. So they allocated 36 machines, and bingo, it was 1.5 months after, we had the whole genome. And we had the whole genome not only once, but we had it 20 times. Okay, okay. And, so this was, obviously there was a lot of, you can say, preparation steps, how to deal with contamination. etc. that I can't go through, where my student did some super clever thinking. But the fact was that, doing the sequencing job that for the first modern human project took 10 years and costed 15 billion kroners, that we did for an ancient degraded DNA from an ancient human in 1.5 months, and it costed 2.5 million. That's what happened, over that time period. So what can you then use this information to? Well, first of all, obviously no one knows a shit about what was these guys, what did they look like, right? You have a tuft of hair, that's all. See, we could use the genome to say something about this person. Well, we could see he's a male, first of all. We could see that he had brown skin. We could see that he had shovel-formed front teeth, that he had brown eyes, that he had dry earwax, which actually increased the risk of getting ear infection. We could also see he had tendency to baldness, so he probably was quite bald. We could also see that he was genetically adapted to living in cold conditions. It's something about how your vessel actually reacts to low temperatures, and that's even though his ancestors or his people had been only in the Arctic region for a very short period of time, so it happened very quickly. We could see that his body stature has been what we called plump in Danish. It would be kind of really, tightly built, for example. So there's a whole number of information. We could see what blood type he had. All that information could get just from looking at the genome. And the only thing you started with, the only thing you knew from that stage was that, well, they apparently had black hair, right? Then what we also could do was to ask ourselves, from where did this guy come? Is he in fact ancestor of present-day Inuits or Eskimos, or is he a Native American, or who is he? And we could compare him then to the DNA of various Eskimos, Native Americans, and Siberian groups. And what we found was this guy has nothing to do with Inuits or Eskimos. This guy has nothing to do with Native Americans. In fact, the closest living relatives of this guy is today found in northeastern Siberia in 3 different populations, that then obviously after that it's split out to 3 different, of one of them being more than 2,000 kilometers away from the Bering Strait today. And what does this show us? Well, this shows us that there must have been an independent migration from the old world to the new world that is independent of the migrations that get raised to present-day Native Americans, and for that matter, get raised to present-day Inuits. It's just that they died out, right? He left no descendants today in the new world, and we can actually also calculate when this migration happened, and it fits perfectly with the age of the first, you can say, tools you find in the Arctic around 5,000 years ago. So this is super cool. And in fact, we also got, which is the same as winning gold in science, the front page of Nature. For the Olympics, I really mean it. It's something that changed your life forever. Okay, so he got this name Inuk because Nature wanted us to give him a name which means human in Greenlandic, I think. I must then admit that when we should make the press release for this thing, which is the one that actually got the biggest press ever in the history of University of Copenhagen. I think on the newspaper side, it has been, the potential readership was like, I think it was 50 million or something, just on the newspaper side. It's crazy, it was everywhere. But we were asked to send I should make a press release together with a press officer, and we were asked, to send it to Greenland for kind of check. They basically they just almost remove everything that had anything to do about his ancestry, because like the Native Americans, they just don't like that he was not a Greenland ancestor, right? Because the consequences is obviously that the Greenland population, the Inuits, hasn't been in their area very long, right? So that was, that wasn't, it's tricky stuff. And what I should mention in the end, and this is completely the end, what are we doing now? Well, we're doing a whole range of things, but what we also are doing is that we are trying to sequence the genome of an historical ancient Aborigine, and these guys here called Andaman Islanders. I don't know if you heard about these guys. It's very, very interesting because it's this group of islands that is lying between India and Australia, and on these islands, the native population are looking like this. They basically look like African pygmies. And this is thousands and thousands of kilometers away from Africa. What is the reason for this? Could these guys become a kind of a remnant of the first migration of humans outside Africa? Or is it a later event, etc.? And really no one knows. And why don't no one knows? Well, because those that are you can reach with these islands are completely intermixed with Indians and English that was there before. And in fact, most of them died out, you know, in contact with white people because of diseases. And then there's a few tribes living in the wild, but if you go out there, well, you can't get permission. It's owned by India. And one of the reasons why you can't get permission is that you will basically be killed. I mean, they're killing people. If a plane crash or something, they're killed. And there's even on the internet, you can find these video footages of the Indian government trying to establish contact, right? So they're coming in the boats and then putting out gifts, and then they're running out, taking the gift and shooting bones and arrows at them, you know. So this is dangerous stuff. But we have access to historical ancient hair samples from them, and this will be certainly political, tense, but also very interesting. Okay, thank you very much.